G'day everyone. We might get a start as we're running a touch late, but I think we've still got a few more people to come through over the next few minutes. Just by way of general introduction and the usual MC sort of stuff, welcome along. This is one in a series of engagement workshops run by the Centre for Mine Site Restoration, basically offering an opportunity for industry regulators, METs and anyone else working in the post-mining space to meet with, hear about and talk to the scientists of the Centre for Mine Site Restoration and about the sort of work that we've been doing over the last 5 years of the life of the centre. Before we get started today, we'd like to acknowledge the Whadjuk Noongar people, traditional custodians of the land on which we gather, and pay our respects to their ancestors and elders past, present and emerging. And there's a couple of housekeeping matters. In the event of emergency, follow curtain people and head out and down. There's a muster point just behind us, I believe, between us and the BHP building. Toilets are located just across here on your left. Both of those doors should be operational, and there's another breakout room just across from us, which you're free and welcome to use at any stage throughout the rest of the day as well. There is another workshop that follows this in the afternoon, and you're welcome to just hang around and utilise that room for working or networking or discussion over the course of the afternoon. The emergency exit is downstairs through the doors that we entered at the opening of the building, and the general now common COVID precautions that if anyone is feeling unwell, please have a chat to the staff at the curtain desk just out in front of us. And if you haven't already, please make sure that you've checked in on the Safe WA app. There's hand sanitizer and all sorts of other wonderful things available to you all. So today we've got hopefully a really interesting and engaging kind of structure. We've got about an hour and a half available to us, and we thought that in our field, being restoration and environmental monitoring and trajectory assessment and determination, one of the most interesting things that we can do is begin linking in people who are undertaking research, hopefully, we think, at the cutting edge of the science, with the people who we are hoping that research is going to be of interest to or will be applicable to or applied by. Now, that is just as important, we think, for regulators and the people who are assessing compliance material coming in or determining the regulations that will bind mining into the future, as it is mining industry and the people that work and consult for them in delivering those compliance outcomes and environmental monitoring. So today we have got a panel of three of our fantastic students that have been working at the centre in the trajectory space. We have Sophie Cross, Mika Vanderheide and Todd Buters. The three of them have a wonderful skill set that ranges across from fauna and the inclusion of fauna in ecological restoration monitoring, eDNA and the use of eDNA in assessing environmental trajectories and biodiversity using a range of substrates, and the use of remote sensing technologies, particularly drones, in environmental monitoring. We also have Paul Neville and myself, who are theme leaders for genetics and trajectory respectively at the centre. And the intent of today is that we will have a bit of an introduction at the start just to give a feel for who we all are, the kind of research that we've been involved in, and how that might apply in the context of the post-mining landscape. And then we'll move to a bit of a, we hope, really engaging question and answer discussion where everyone will have the opportunity to talk both among yourselves and to us, ask questions, work through where the science is currently being applied, where it can be applied better, and where the particular knowledge gaps and opportunities currently lie. So by way of very brief introduction before I hand over to Sophie and Todd first, and then to Mika and Paul to talk to eDNA and the use of genetics in monitoring, I presume that everyone is pretty much across the concept of trajectory, being essentially the path that an ecosystem takes from a point at which we begin, or actually a long time prior to, but when we begin restoration, through to some unspecified point in the future where we have achieved some form of goal or aspiration or target for that system. Now, trajectory is obviously not a a concept that is unique to ecology. If we think of the trajectory of a ball, for example, going through the air after it's been kicked, you can have really subtle impacts on the trajectory of that ball, the path that it takes. For example, a gust of wind can alter the direction or the height or the speed at which the ball is traveling. Or we can have some really overt impacts on trajectory, like the ball hitting a brick wall and stopping completely. Exactly the same thing happens with ecosystems, and there are a lot of different points along that trajectory where we can deliberately influence the outcome or the attainment of those goals and targets that that ecosystem has in the long term. And why trajectory essentially underpins rehabilitation, restoration, and everything that we do in the sort of environmental post-mining space is that everything that we do, from planning to establishing a landform to direct seeding to monitoring, is aimed at ensuring that that landform attains a certain level of functioning or a certain number of characteristics, whether that's a biodiversity target or whatever the particular outcome may be. And if we can identify and know where to make changes in that system and along its trajectory, and where to look to see whether those changes have been successful in altering the characteristics that we desire, then we can report earlier and have a much greater confidence that that system is actually going in a desirable trajectory, in a direction that we really want it to. So by way of just kicking things off, I'll ask Sophie, Dr. Sophie Cross, to come up and just introduce herself and her background in research, and the specific context of which she undertook her PhD and postdoc, which is the behaviour of fauna in the post-mining landscape.
Hi, so I'm Sophie. I finished my PhD early last year at Curtin University, and my PhD was broadly looking at how animals respond to mine site restorations. So in assessments of habitat restoration success, monitoring animals is pretty uncommon compared to monitoring vegetation. People kind of just assume that they'll come back following the return of vegetation, but in practice it's actually very difficult to recover these functioning ecosystems with the same community dynamics and behaviours of the animals as they were in their undisturbed states. So my PhD broadly had two components. The first was looking at how animal communities in general respond to habitat restoration. So this was using things like camera traps in the bush, looking at differences between reference and restored vegetation, seeing if the similar community dynamics and species were coming back into the restored habitats as were present in the reference areas. And then I started specifically looking at the behavioural responses of monitor lizards. And monitor lizards are, I'm pretty biased, but I think they're pretty much the coolest things in Australia. Basically, they have the largest size range of any vertebrate taxa ever. They basically range from about 20 centimetres all the way to 3 metres in length. And this presents a really interesting group to kind of look at how changes over multiple spatial scales impact animals. So you've got the small species which have relatively small home ranges, which probably be more impacted by habitat change, and the larger species which are probably more accommodating to this change that can roam over various different habitats. So the latest parts of my PhD look specifically at how these monitor lizards behaviourally respond to this habitat change. So I use things like VHF tracking specifically to look at where they were going in the landscape, how long they spend in particular areas, whether there are other parts that they completely avoided, and the potential costs associated with using different parts of the landscape. So particularly for reptiles, which are, they regulate their body temperature with the surrounding environment. So things like heat are very likely to have quite strong consequences for animals. So if there's an open and homogeneous landscape, they're more likely to have increased temperature costs associated with using these landscapes. And early stage habitat restoration can present potential costs because the vegetation hasn't yet established, so this might impact their movement through these landscapes. So specifically after looking at individual movements, I kind of looked at the population responses as well, looking at how they move through the landscape using tracks and diggings and burrows and that kind of stuff as measures of their movements.
Thanks, Soph. One of the key things that is underpinning in trajectory is having some sort of a baseline to begin from. And what sets aside restoration, ecological restoration, from rehabilitation and other activities is the utility of a reference ecosystem. You can't determine how a system has progressed along a trajectory towards an end point if you have no idea what characteristics you want that ecosystem to have in regard to a predetermined goal or target. And the next person that I'll ask to come up is Todd, who's undertaken his master's in the utility of drones in restoration monitoring and environmental monitoring generally. And the key sort of tenet of some of that work was how you actually assess the characteristics of a baseline in the reference ecosystem, and then how you can compare that over time series in restoration.
Thank you, Adam. It's nice to see everyone here today. As Adam said, my name is Todd. I did a master's on drone-based monitoring with the CMSR. I finished about a year and a half ago now. And a little bit of detail on what I did. I was looking at using drones. Unfortunately, nothing as fancy as this. This is a relatively recent purchase to monitor basically the process that a system goes through as it's returning to its ideal baseline. And the way that we did that was by having a essentially sample ecosystem that we were monitoring. This was based out at the Shenton Field Station, and we were just flying over it every day, getting pictures with both a regular camera and a multispectral camera, which is, I'm not sure if anyone here is familiar, but it's a special sort of camera that's used for monitoring plant health. It uses light in the non-visible part of the spectrum in order to assess that and hopefully give us a little bit of extra early warning if something is going wrong. And we use those two cameras to monitor the system that we had set out and pick out and count the plants that we were interested in. We had a target species and we had a basic backdrop of weeds, and we were interested in picking out the target species from the weeds. We were pretty successful with that, and so we've purchased this drone here with a better sensor, and this is getting some really good results as well. So far, unfortunately, we've been interrupted by COVID. We've had a couple of trips and experiments that we had planned that were all pretty much early to mid 2020, and obviously I'm sure you can all imagine what happened with that. But we've still gotten it out there and gotten some good results. We've done comparisons of drone-based and ground-based assessments out at the Hanson site in Ellenbrook, and we've had some really good success with picking out specific plants there as well. Most notably, we've been able to pick out just the flowering cones of Banksia trees and get a count on how many flowering cones there are, which is a pretty amazing thing, just to pick out those cones from a drone that was flying, 60 metres in the air, so it's pretty far away, still picking it out. We're further broadening our range with a couple of extra things that we've got in the works at the moment. So one of those is detection of, it originally started as large mammals, but we're finding that we're detecting large mammals, small mammals, birds, basically anything warm-blooded. We're working with a thermal camera to try and detect it. That's with a bigger drone, which isn't here, unfortunately, because it's rather large, but we've had very good success with detecting things like foxes and kangaroos in quite thick bush. Despite the crown coverage, we're able to pick up the thermal signature and then switch over to a standard visual camera and identify exactly what it is we're looking at, which is quite an interesting job, actually. We've had some surprising results. We've had the disappointment of switching to visual and finding out that that hotspot was actually just a metal barrel that was sitting in the sun. We've also had some nice surprises where we saw a perfectly circular hotspot and thought, that'll be the lid of the barrel. And it was actually a fox curled up for a sleep, which was quite interesting. But we're getting some really good results. We're working with automating the process of picking certain things out from the environment, which I've got set up on the computer here. I should probably hand back to Adam, otherwise I'll be standing here talking about it all day. But if you're interested in what we're doing, come up to me afterwards and we can have a chat and I'll show you some of the things that we're doing.
Thanks, Todd. And there'll be plenty of opportunity, I'm sure, to have Todd or Sophie or myself or Mika or Paul chew your ear off with regard any of the particular research outcomes. As probably everyone is aware, historically, monitoring essentially has comprised people walking landscapes on foot and eyeballing vegetation communities either to get presence, absence of species or some other metric like cover. Not always particularly useful, and in the context of determining the functioning of an ecosystem, almost completely useless. So there's been a great push in the last few years to broaden the scope of taxa that are incorporated and in one pass determine much more from data sets that are available to us, which is where eDNA and Mika and Paul's work comes in.
Great, thanks Adam. G'day everyone. So today Mika and I will be just talking to you briefly, giving you a bit of a refresher course on what eDNA actually is, and then just some of the general findings from our work and where it could be applied in the future in your business. So one of the constant themes of the DNA node of CMSR is that advancing technologies have revolutionised the data that we can collect and also the application of that data. And that's where eDNA-based monitoring has come in. This is something that you just couldn't do 20 years ago, but because of huge changes in DNA sequencing technologies and associated analysis methods, we can do it now. So eDNA 101, there's quite a few people here that I don't think have been to any of these CMSR presentations before. So just a bit of a introduction to eDNA. What is it? It's just organismal DNA that can be found in the environment. You can pick it up in soil, water, on plant surfaces, etc. Where does it come from? So everyone's shedding DNA into their environment. Here, you're shedding DNA into the environment from your skin cells or your hair, and organisms shed it through dying, defecating, urinating, mucus, scales, etc., being rubbed off their bodies. So how can it be used? We can use it to monitor ecosystem changes, obviously. We can also use it to detect invasive species or to really help us monitor threatened conservation priority species. So the workflow here, samples are collected. Up the top, as in traditional ecological monitoring, then we pretty much mash them up and extract DNA from those samples. And then we use a molecular magnet to pull out DNA sequences that we're interested in, or barcodes. So humans have a unique barcode sequence, an aardvark has one, an elephant has one, Acacia saligna has one. So we can pull out those unique DNA sequences with molecular magnets as an analogy, and we can use different magnets for plants, for vertebrates, for invertebrates, etc. And then we blast those sequences against a database of known sequences, and it'll tell us what's in our sample of scat or water. And yeah, we can then further analyse that data just as you would traditional ecological data a lot of the time. So this has been really extensively used in marine settings. It's really commonplace now. I don't think there's anyone from oil and gas here, but it's a standard monitoring tool. Terrestrial monitoring is probably lagging behind a little bit, I'd say, but rapidly catching up. So some of the advantages of it can be gathered quickly, readily analysed and verified by third parties. So it minimises the challenges of some morphological approaches. Mika will be talking about stygofauna later on. Stygofauna is really hard to survey, as you know. Ground-dwelling arthropods are really difficult to survey, as you know, because there's just not a lot of people with expertise in looking at them. So we can also monitor without having DNA sequences that we know exactly what they are. They don't exactly have to match a species in the database. They can match a family or genus. So we can get an idea of what's going on even if the reference database is incomplete. So what we've been pushing the whole time with the CMSR is that this is a more holistic model of mine site monitoring, and it's well and truly overdue. So we want to look at everything from soil microbes to mammals as part of this monitoring approach. That's a whole ecosystem monitoring, not just looking at plants and then inferring what's going on with other parts of the ecosystem. So obviously fauna, Sophie talked about fauna as being really important and often being ignored in restoration monitoring. They're obviously really important, and we can get at fauna monitoring quite quickly by using eDNA. So some of the deliverables from Mika's PhD over the last 4 years, we've done extensive development, field testing. Mika's writing industry guidelines now for eDNA-based biodiversity assessment. And we've also looked at soil microbial communities, which are pretty difficult to study in other ways, but quite easy to study using eDNA. And we've also developed guidelines for topsoil management from those studies. So one of the key messages we've tried to convey right through the last 5 years is that eDNA is not the silver bullet. It's not going to solve all your problems, but it will solve some of them. So further development of assays, sample design, looking at DNA degradation and sensitivity are needed, and also populating those databases if getting down to a species identification is important to your business. So one of the issues, I'll just go through a couple of issues. Determining absolute population abundance from DNA is probably never going to happen because different organisms shed DNA into the environment at different rates, and there's a whole heap of other reasons why. So whether you've got 1 qual, 4 or 8 at your site is probably a difficult question to get at, but there are approaches now that are being tested that will enable that somewhat. We're getting better at it. How to ascertain ecological functionality is a difficult thing to get at with eDNA, but Mika will talk a little bit about that later on, and we are getting closer to doing that. So, you know, my message is that better tools are needed for monitoring. eDNA will be not the ideal tool in some situations. In some situations like monitoring stygofauna, it's probably the best tool that you could use. But in most situations, it's probably a combination of traditional monitoring techniques and eDNA that are going to work best for you. So I'll just hand over to Mika now.
Thank you. Thank you very much. Okay, yes, so as a little project overview of my PhD, I started by looking at a bunch of different substrates that we could use. And then I went into chronosequences of restoration at 3 different locations in WA, just to see how we could look at the recovery of soil microbial communities, because they're an established target of DNA metabarcoding. They're the functional basis of ecosystems, and they respond very quickly to change. So it's a good way to get a snapshot of the ecosystem. I tried looking at vertebrate diversity using pooled Scat samples, and finally looking at invertebrate recovery. So this is applying the DNA metabarcoding to well-known indicators of restoration success and environmental condition. Oh, wrong way, sorry. So I went to the Swan Coastal Plain, the Jarrah Forest and the Pilbara, and I collected scat, insects from vein traps and pitfall traps, and soil samples to get all the things I needed. At each of those locations, I also had 2 separate reference sites that were sort of north and south of the restoration sites, just to make sure that any patterns that I was detecting weren't influenced by the distance to the reference site, because I'm looking at the similarity to reference sites over time, and I want to make sure it's not just because they're further away that they're more different. So just as a general, the take-home messages that I got from this is that overall, one size does not fit all for a few reasons. This is the results of my soil study, my soil microbial study. You can see there's the effect of the restoration method here. In the yellow, you see the Swan Coastal Plain, which is the site that we had direct transfer of topsoil. And you can see how that's affected the bacterial results. So the youngest and the oldest sites are the most similar to the reference communities, and then you have that sort of dip in the middle. Whereas at the other locations, you can see an increase in similarity towards the reference communities that you would expect in a trajectory towards those communities. There's also a difference between the bacteria and the fungi, and this can be attributed to ecological things. For example, the Jarrah forest has that very clear trajectory in the fungi. It also has these very large mycorrhizal networks in the forest compared to, for example, in the Pilbara. So you have to keep your restoration methods and your ecology in mind when you're sort of designing your eDNA surveys. Once again, ecology is very important. So this is just the invertebrate communities. At the top is the pitfall traps, and the bottom is the vein traps. And you can see you get these very clear trajectories in the Swan Coastal Plain and the Jarrah Forest. In the pitfall traps more than the vein traps, and that's because pitfall traps will catch things closer, whereas vein traps have a higher dispersal of the things that they catch, so they can be catching things from outside your study area. Pilbara had a very different result, and again, we attributed this to the ecology of the Pilbara. Pilbara already has a lot of open area in the reference and the restoration communities, so you may not be getting those differences in the invertebrate communities. Finally, your different locations will also affect which substrates are suitable. This is the results of the Scat assay study that I did, and for this I did pooled Scat samples from each of the different restoration sites. And you can see that we got a lot more birds from the Pilbara, barely any from the Jarrah or the Swan Coastal Plain, and this is basically due to Scat detectability and persistence. It is much easier to see bird Scat in the Pilbara on that red rocky soil than it is in more vegetated areas with higher rainfall where it could just wash away. We've been having a little bit of trouble getting vertebrate DNA from a lot of things. I've tried with soil, but it tends to be degraded too fast, so I don't get a lot unless the density of vertebrates is really high. We've had some luck looking at flies instead, carrion flies, getting stuff from feces and such, and also from sediment hollows. So those tend to have quite a lot of the vertebrate diversity, possibly because it's protected from the UV radiation in the sediment hollows and their high traffic areas. So one of the things that eDNA is pretty useful for is getting that additional interaction type data. So from soil, you can get trophic mode of the fungal community, you can get the metabolism of the bacteria through third-party annotation and such. From Scat, you can get diet, so if you're interested in a particular animal and its restoration or translocation, you can use the Scat to figure out what it eats to make sure it's there. From the invertebrates, you can also get the associated plant communities, like the plant communities that they're ingesting or pollinating, and that can help you understand the interactions between different organisms in your restoration. And that can be difficult to get from some other traditional methods. However, eDNA will always be another tool in the toolbox, not meant to like replace everything else, but another tool that can be very useful in certain situations. So as a final, trying to bring it all together, I've been trying to outline how you would design an eDNA survey for restoration. And from all of my studies, it's clear that you have to really understand the environment and the ecology of that environment. That's sort of a filter that will determine which substrates are suitable and what you can get from those substrates. And that will help you figure out what sort of assay you need to use on those substrates, whether you want to look at invertebrates and their associated plant communities, or you want to target a specific animal and want to find out their diet and if their diet changes in their different restoration. Finally, you can use all that to compare your restoration and your reference databases to try to figure out what thresholds you should be looking at, whether that's the similarity to the reference communities or how much of the reference taxa do you need to be able to consider that an appropriate threshold. So finally, after all of this, what I'm working on now is subterranean fauna, and it's a very exciting field because we're basically just picking up water from these holes and then seeing all these wonderful things that are inside and comparing it to our net holes and what we get from those. One thing that's very cool about this project is that we're creating our reference databases so that we can get very accurate identifications of all of these. And I'm very excited about the rest of this project. Anyway, thank you.
Great, thanks, Mika. So now it's over to you for, say, 15 minutes. I'm not sure what time it is now. What time is it? 22. So at 5:02, we'll get back together. On your table, we've got some paper and texters. So if you're on a table on your own, maybe move to another table. And have a go at answering these 3 questions here, and then we'll get together and just discuss your responses to them. And just put your business next to your answers, you know, what company you're from. And yeah, go for it.
Maybe industry rather than company, if that's something you don't want to include on there. I should note as well, and just in the interests of embarrassing her as much as anything, there's also one of our PhD students here who is still currently studying in Holly Bradley over here, who will shortly be joining Sophie and Mika and the others as a doctor. And Holly, Mika, Sophie and Todd will all be sort of orbiting around during this 15 minutes for questions and discussion if you want to talk to anyone. Not that.
What's the point?
I need to make it fill first. Or we'll get stuck into the discussion going.
That's future.
Vanessa. Vanessa. Am I able to have you wander around as well for questions and tables? I'll cover the first half if you like. Yeah. Right, for the last half an hour, we might just delve into a bit of a chat and allow everyone the opportunity to talk to our naughty school children up the front. Everyone's got like a bashed look on. With regard the three questions that have been asked, there's obviously a great deal more complexity to trajectory and ecological monitoring and everything to do with determining success and pathways towards closure in the post-mining landscape. So there's an opportunity after this as well to have a conversation with any of us here and ask some more specific questions. Everyone's going to come with a particular suite of interests. It sounds like there's a particular Steiger fauna group at the back that probably wants to have a chat to Mika longer term. But just to start off with, the first question regarding the current application of monitoring data, whether that's remote sensing, eDNA, or other even existing sort of foot-based methodologies. Is there any group that wants to kick us off just with an example of that current application of the technologies that we've discussed today? Particularly interesting or novel application? Sorry, I meant to say currently.
So has anyone used eDNA or drone-based monitoring so far? And what are your experiences with it? Yeah, Fern. Thanks, Fern.
We've certainly looked at drone monitoring within our operations at the moment. Probably the technology probably isn't there for the detail we require on the ground with the number of species we see. Certainly with some of the eDNA stuff, well, we've done a fair bit of DNA stuff, both with Kings Park and also the centre over a period of time. So I guess it's a trajectory we think is important to us. And I was interested in some of the stuff around that we spoke about with the soil fungi that you raised in your presentation, and also bacteria as well. So we spoke about that just recently. So, but I think these technologies will keep continuing to improve, and I think hopefully will help drive some costs down for our business as well into the future.
For other industry that are here, is there an actual approach to determining, say, microbial diversity at any sites? If eDNA is not being used, is there any interest in that space, or has that been undertaken to date?
Well, I'm from Mount Gibson. We have a number of mine closure plans, none of which contain any performance criteria relating to the health of the soil structure itself. It's assumed in that it will then account for much of what you see above ground in terms of richness and biomass of the vegetation. So for us, it's a blind spot. I'm not blaming the MCPs. We only know what we knew years ago when we put them together. I think it's a really interesting and powerful thing to do. It'll enrich that space of understanding of ecosystem function. So we don't do it. We probably would like to. And Paul, I think if there's a grant that gets up, we'll have a bit of technology that can help with that.
There's another question as well.
Yeah, I suppose to touch on that point is where I suppose definitely as an industry, I think evolving in that place. I've come from baseline soil surveys that essentially it's been a presence or absence of if cryptograms present. That's it, you know. And thinking that from a soil health perspective that it's from a good baseline. But as we go through, I suppose, topsoil assessments and before we spread them, you know, the microbial aspect is, it's definitely something that we need to get better at. And I suppose looking through that DNA perspective that we can understand the viability of our topsoil before it's spread and then as it moves through that, because that's really, I suppose, ultimately the foundations of it. I think in general, the industry is, I suppose, trying to evolve and work better to understand from a holistic approach as opposed to the absence, presence of vegetation in a quadrant perspective. You know, looking at, you could have the old traditional approach of that it's absence, presence, and you get your 70% species richness back, but it's not the whole story. So it's looking at that whole landform. And by looking at the UAV's perspective, we can look at an entire landform and understand erosion and and that sedimentation was processed in the landform evolution process, as well as then as a vegetation establishes. And we've been doing some good work with Paul and his team around plant pollinator eDNA studies. It's, at reference sites that have, pristine environments at this stage, so that we can look to see if we can introduce it into the restoration component. But yeah, there's lots of work that I suppose needs to be done in the space.
Just to go back to the soil microbial communities and the soil health part. Part of the thing that's quite useful about using the DNA metabarcoding or even shotgun sequencing is you can get functional information from that, either through third-party annotation from the identification of your microbes or through functional genomics. You're looking for functional genes in the soil. So for example, for my soil study, I was looking at bacteria metabolism, and I could see that there was more organic matter decomposition type bacteria in reference soils and more naphthalene degraders, for example, in the restoration soils. And similar for the fungi, trophic modes were different between the restoration and the reference. You had more saprotrophs, so those decomposer fungi, and you more symbiotrophs, so the symbiotic fungi in the reference, compared to more pathotrophs, so more pathogenic fungi in the restoration. So it's good to get that additional functional information, and I think we should definitely be pushing for that and not just lists of species that were present and absent.
I'd just like to add that I would like not to see things like the soil fungi as a completion criteria listed as the end goal, but the techniques will be amazing, but more as an analytical tool. That if we've done a certain rehab protocol on a number of sites and one performs well, one hasn't performed well, then that it's an analytical tool to see, what's worked, what hasn't. But being from industry, it just makes you, everyone's a bit anti the list of plants and whatnot, but, we can only, we've gone through that process with land function analysis, having a million things to monitor to get a result, when a few of the, the plants, as you were saying, does reflect what's there before. So we don't want, 50 things that are suddenly in our closure plans that we must comply with to get sign-off, relinquishment, which is what the aim for industry will be, but to use them as an analytical tool to better our outcomes, absolutely. But more, that's
has gone wrong. What do we do?
Absolutely, yeah. I mean, who looks at soil bacteria? It's not something any of us mention. So it's very exciting that you have two sites and you've done exactly the same things. You think the soil's the same, but it could be, well, they've done well here and not there.
I think Mika's point that it's a one tool within a toolbox is incredibly important. And I think the one message that the CMSR is pushing in terms of trajectory generally is that no one of these approaches is going to be particularly groundbreaking to answer every question at every site. There's always going to have to be a selection or a suite of these tools that are applied in tandem. But to that point, one term that is increasingly coming through, whether it's in mine closure plans or guidance or even just the literature, is this concept of resilient, self-sustaining and functional in terms of the post-mining landform. And a lot of these tools allow determination or empirical evidence to support that resilience or the self-sustainability or the functional, whereas the sort of traditional assessments of cover or presence-absence really give no indication towards any of those three functions. So you can't talk to whether a species is recruiting as it would in the pre-disturbance system or whether fauna are behaving differently in that system. And I might just ask Soph to give a quick discussion of what she saw with regard to, for example, the varanid goannas, utilising restoration and rehabilitation areas slightly differently from the surrounding landscape in that regard.
So I had two main studies which looked at the behavioural responses. One was looking at community responses, so that was in terms of walking through reference and restored landscapes to see if I could pick up signs of tracks or diggings or burrows and that sort of thing, and then judging how old they may be as well. So for this study, it was interesting because we saw that while restored landscapes were being used, the use of this seemed to be more opportunistic and by larger animals, so that the larger monitor lizards are more able to withstand the temperature fluctuations, and they kind of, but even though these animals were using the landscape, it seemed to be pretty much to just pass through these areas, because we didn't really record much in the way of diggings or burrows or that sort of thing, and there was very, very few signs that these animals were using these areas long term. But interestingly, when I started looking at how individual goannas move, so I stuck a tracker to the back of a young adult parenti, which is Australia's largest lizard species, super, super cool animals, again, very biased. But when I started to see how that animal moved through the landscape, we started to see that restored landscapes were actually facilitating some level of long-term use for these animals. So while my study showed that the animals spent a lot more time kind of moving slowly through the reference landscape and kind of using overnight burrows, not necessarily having to return to one core area for the thermoregulation and refuge, in the restored landscape, the animal did still have that, you know, fast, opportunistic movement across the landscape, but there was one core area of that restored landscape that actually it was using as a burrow, and it frequently returned to this area, presumably to regulate its body temperature between foraging trips. And probably one of the coolest things of this study was an opportunistic sighting of this animal actually mating with another parenti in the restored landscape right next to the burrow. So while we might be seeing that community responses not really coming back into these landscapes quite yet, and you know, might on the face of it seem that these areas are not really supporting that long-term use. When we start to look at individual behaviours, we can see that these landscapes may actually be promoting these longer signs of use, but not specifically by, you know, the whole range of species. It might just be more suited to the larger species and species that can withstand these temperature fluctuations, or might have less chances of being predated upon because they can move through these landscapes without fear of, you know, a predatory bird coming along and pick them off or something like that.
One of the things I think we've focused on sort of large-scale monitoring here, but one of the key areas that I think eDNA can be useful is the study of conservation priority species, you know, understanding their ecology, their interactions with other organisms, their diet, and also getting a handle on distributions. For example, we're looking at sampling NAMA holes now and bore troughs across really large-scale areas, and looking for things like, you know, rare parrots, etc. So I think that is will be a big area for use of this technology.
Maybe with regard to the second question, has anyone got particular examples or areas where they feel that these technologies, particularly maybe remote sensing, are being underutilized, or there are particular issues with their application?
I guess what questions do we need to solve for you guys before it'll be operational?
I suppose this isn't a question to the regulators, it's more, or I mean to the researchers, it's more to the regulators in the room. I feel there is a big engagement from the industry, but every proposal I put in front of the GM, my GM and my business is, well, what will this mean for us? Will we get faster approvals? Will we get to closure? Will we get to relinquishment faster? It's fine to do this, but will it solve our problems? And so I suppose it's a partnership between industry, research and government and regulators together of how we can go on this journey and ultimately to get from a mining house perspective to closure and relinquishment, and ensuring that, you know, from a risk perspective, the more data and I suppose putting those pieces of the puzzle together, that holistic approach, that we could really get to the end game together. Otherwise, mining houses won't do it, really.
that's something that's underpinned a lot of the research, particularly in the fauna and drone monitoring sense, where we're talking about cost efficiency improvements, where rather than having boots on the ground, you might be able to undertake remote sensing monitoring. Maybe Todd, if you can talk for a minute or two about the utility of a one-pass solution, for example, in the drone-based remote sensing and what that can offer.
Okay, so basically a little bit of background to this. When I first started my master's, the first thing I did was a literature review of drone use in restoration. We noted a couple of issues that were pretty prevalent throughout all the studies that we assessed, and one of those issues was that in general, people would be flying over and assessing a single thing. So it might be, you know, assessing what the hydrological situation was by generating a digital terrain model and working off that, or looking at what sort of percentage of plant cover you had, or, you know, looking for one particular plant. Whereas with drones, particularly if you're using multiple sensors, you can fly over once and get enough information to look at several different things. So one of the things that we're interested in doing is not just looking at, for example, with our test that we did during my master's, not looking at just identifying the plants, but we also had a multispectral camera to help monitor their health as well. So we ended up proposing a one-pass solution, which revolves around the use of multiple sensors, all mounted on the same drone. You fly it over once, and you get enough information to get, I believe at the time I said, a complete assessment of environmental health. I was a little bit arrogant at the time. I thought I'd proposed the solution for everything. Obviously, that's not the case. But just as an example, if you're flying over with RGB, multispectral and thermal, you can assess plant cover, you can assess plant health. You can get a little bit more information about the plant health with the thermal, because with the multispectral, I should say, you get an idea of whether the plants are healthy or not, but you don't know what is causing the issue. So the addition of thermal to that lets you determine whether or not it's water stress. The thermal will also help you detect the large, or as I said before, not so large warm-blooded animals. So it's really about maximising the data that you get from a single pass and making sure that you're actually using all that information that you get, and not just gathering a huge amount of information and using it to say, there's 3 banksias in there.
I think the key thing there from the industry perspective is you would otherwise have to have someone go out and do an erosional or landform stability survey, a fauna survey, a flora survey, a weed survey. All of these are different personnel with different costs, whereas the potential application for that drone-based remote sensing in the future is just one operator, one drone, and probably a couple of days in the field, and then some analytics back in the lab. Maybe a question then directed to the regulators, our regulator table. Where do you think that in future these technologies might assist in either compliance reporting or in the assessment of information coming to you?
I've just been listening to that and thinking about where it would really assist early indicators. It seems like a lot of the time it's a monitoring at the very end and showing, well, it hasn't met what it was meant to, but there's nothing we can do about it now. And I was thinking about also your comments about not having all these different things to monitor, and it would almost be if you had something really early on that might not be the end target, but would tell you if it's trending in the right direction or you really need to do something now before it's 10 years down the track, that would actually help it get there. That would actually really help guide us and industry, because I don't think we're actually monitoring trajectory. We don't actually have an idea about the path things should go. There might be an end target, there might not, in lots of cases, be really defined. And there isn't a good understanding of, is it trending in the right direction or not? I don't see that from this information I get. It's like, it looks okay, or it's like, it hasn't met the target yet, but there's no indication from what I'm seeing about, like, is it trending? I don't know if you get the same.
looking at potentially adding in milestones when we're looking at trajectory, because it gets set at the start, you get to the end. If it hasn't met what it was supposed to, it's just too late. So putting in milestones as you're going along that you can meet, that make sure you're on the right track, are going to be really helpful.
I think there's like hope that there would be an understanding, but unless it's actually required, and lots of times people can't justify doing it. Like it might be, this would help us get an understanding, but if it's not stated there, it's not done. So I don't know how to balance that so much.
And that comes back to a point here about the assumption of a lot of functionality or biodiversity returning to systems, and this offers, or trajectory monitoring generally, offers an opportunity to look at the divergence along that trajectory and flag early on where you might need to intervene. There was another question?
Yeah, look, I think you've got to remember here that the problems haven't changed. The way the data that we're looking at hasn't changed. When 30 years ago I was looking at organic material in rehabilitation, I was trying to move my hands together and we monitored on how dark your hands were. We're still monitoring the same thing. It's just the technology that's changed. So, and trajectory is just a trajectory, it's just a path forward. So this is not a panacea. These are just different ways of collecting data and more efficient ways, and the cost and safety issues that have driven that over time. The thing here for me is it's our understanding of the data that needs to change, right? Trajectory is just an estimate of a correlation between the data that we're expecting to see. It's not an actual thing. So the variance around that trajectory is what's important. How much variance are we seeing, how that's changing over time. We talked at this table about in mineral processing, we used to look at total quality management. Same sort of thing, you'd be looking at a trajectory over time in terms of quality and starting to understand that variance, and if things varied above or below the line 3 or 4 times, when that was an indicator that you needed to interject and intervene and make some sort of change. This process is absolutely no different. I think we just got to remember that it's about understanding the data and looking at the data and understanding that variance, and applying analytical methodologies around that data to understand it. And that's how we'll sort of start to get to that point of compliance or relinquishment or whatever it is that we're trying to look at. It's not, the technology is not going to allow us to do that.
I think that's a good point, and something for the scientists that we need to be constantly aware of is that we're looking at developing tools that increase the maybe accuracy or precision of information that's coming back. But at the end of the day, any ecosystem, particularly here in the Southwest or the Midwest or somewhere like that, where we've got these ancient ecosystems, we're talking about relinquishing or closing or releasing sites, giving them onto other landowners within, say, 5, 7, 10 years of establishing that rehabilitation restoration process. But the time period to actually fully restore that system or to return all of these processes might take 100 or 500 years. So it's about generating enough confidence in the information that's been provided that we can forward plan and actually have a level of understanding that that trajectory is going to retain on a favourable path. Paul, do you want to have other comments?
Just talking about that and what Vern was mentioning, if you remember the inverted bell curve that I had in the Swan Coastal Plain, if I didn't have those older sites, you would assume it's just crashing. So sometimes it's important to have those long-term monitoring just to get an idea of what kind of trajectory you might expect to happen. And so you don't cut it off too early at 5, 10 years and be like, well, you had a good trajectory up to there, but then it hit a brick wall.
My perspective on trajectory is really driven by the process, right, that you start with. So I don't think in short-term mining projects you're going to get a trajectory of any confidence when there isn't the background work that's been done. I think the process for, say, actual woodland is the critical part about making sure the process is done day one to the best of the knowledge at that point in time. And with long-term monitoring via these sort of techniques, the trajectory will be known. And I guess using that knowledge over long-term research, I guess, across numerous different sort of ecosystems, the process should be driven by the regulators to make sure that that process is followed with the best information at that time, because we know the trajectory will follow because of that long-term research. Does that make sense? Because a lot of projects are 1, 2, 3 years in terms of length, particularly in the gold fields. So it's important to make sure the process is right in terms of restoration, and the trajectory will follow, because trajectories, like was just discussed then, is over a long period of time to understand what's happening. Correct. And what Mika's saying, what we've learnt is, over 25 years, what we thought wasn't a good technique, the trajectory has changed over time with different techniques, and now we use a completely different method to what we thought early.
So for the industry in the room then, what is the kind of number one thing that is a constraint or a limitation towards that closure and relinquishment process, where some form of monitoring would allow a quicker or a more cost-effective or a better approach for that for you?
You mentioned the baseline, Adam. You're doing a waste dump. There isn't a baseline because we go to the environments and have to have research through time, as Vern was saying, maybe other mine sites, because a gold mine might be up and gone in 5 years, so it's that. But that would be the initial sticking point before we have that background, that research that we're dealing with quite often. The challenging sites or mine site rehabilitation is your unique landforms that you're creating. You can't have a baseline because there is no baseline.
Yeah, that's a particular point of interest for me. My research over the last 7 years has been in magnetite tailings, and the closest baseline geochemically to magnetite tailings is moon regolith, compared to a comparison with local natural topsoils. So no, there is no reference system in terms of that soil chemistry locally. But there are still inferences that can be drawn from other systems, other ecosystems in the region, other communities, other soil types and structures that can maybe guide those processes. And I think that highlights one of the key issues that we certainly face in the science, and maybe is also a regulatory issue as well, where obviously the imperative from the mining industry's perspective is to get the minerals out of the ground, to make the profit, and then to be able to relinquish the site and move on to the next site. Not cynically speaking, just talking from the business perspective. Whereas our perspective as ecologists, and more broadly the kind of societal expectation, is that that land will be returned to something that is of benefit to the surrounding landscape at the end. So I would counter that argument by saying mining industry in that situation should have an expectation around what the substrate is going to be created by that operation, the chemical, the biological, the edaphic characteristics of that waste rock, so they can plan in advance the targets and the goals for that rehabilitation and restoration process longer term, rather than saying there is no reference.
Absolutely. That's what coach planning the statutory guidelines follow, but I was just referring back to your statement, on baseline, it is something to be remembered. And you said, yes, certainly you can make inferences to other things, but they're inferences that won't be solidified until we do the research, and that takes time.
And maybe that's a great point again for the kind of eDNA space, where you can have an expectation of how you would see communities assemble naturally after maybe a catastrophic natural disturbance like glaciation or a volcanic eruption or a landslide. You would understand how you might expect that system to recolonize and reassemble over time. And you can use these techniques and technologies to quantify whether or not you're seeing that same recovery or a different recovery, and maybe identify areas where you can intervene in that system if you're getting a divergence from what you were expecting. Other points there? Are there any direct? Yep.
I've just got a question for Troy, actually. Yeah, your Darwinia and your Lepidosperma, you have to prove that they're self-sustaining plant communities, is that right? Yeah, so could you see either of these technologies being used to demonstrate that?
Currently, we re-established, we're counting seeds in re-established soil seed bank. So eDNA is a much easier way of doing that rather than having consultants on their hands and knees dusting up soil from plots where we've planted replants. So absolutely. I think eDNA helps with Steiger fauna as well, just getting a very strong positive or negative sort of issue response. But coming back to the DRF, I think eDNA will be very powerful ultimately once those... We took 1,300 Darwinia plants and we currently have 4,000 of them growing in even rehab landscape. So we think with time we will just see them continue to grow, throw seed, and that seed then migrate by virtue of the ant population, sort of marching out around the waste dumps. So we can do it the old-fashioned way and count them manually, but we might use some of these more modern techniques. So as soon as I get your proposal, Paul, we'll be looking at doing a purchase order. So thanks for putting me on the spot.
So I suppose the limitation there for some of those approaches is whether or not you can glean enough information from the eDNA to look at abundance as well as presence-absence. Is that something that is a future pathway for that technology?
Yeah, I think I'll just chip in for a sec. Abundance, obviously, if you have a plot where you're surveying and you pick up 20 detections of chudich in your samples and you have another plot where you only pick up one detection of chudich in your 20 samples, then you could pretty confidently say, I think that there's more chudich in one site compared to another. But whether we're going to get beyond that, I'm not sure, but there's a lot of studies coming out at the moment where people are trying to do that.
Yeah, so part of how it works is because organisms have their own different DNA deposition rates and all that, it doesn't work very well if you're looking at communities for abundance, even with relative abundance, because the bigger organisms tend to shed more DNA than the smaller ones. And mammals tend to shed more DNA than reptiles. That. But if you're looking at specific target taxa, for example, a conservation priority species or something, you can get sort of relative abundance measures by the number of copies of DNA in, for example, a liter of water or something, if you're comparing just that target taxa with itself rather than between that target taxa and different species.
And just going back to a point I made in the presentation, I think that the eDNA technology has an incredibly powerful ability to look at change in ecosystems and collect that baseline data. Some of the work we're doing with MinRes, where we're looking at pollinator communities at an undisturbed site, that would just never happen pre-mining, pre-approvals. No one would ever collect that information, I don't think. So we can look at a whole suite of conservation priority species just by taking flower samples off them, squishing them and sequencing, and we can identify what pollinators are interacting with those, or what insects are interacting with those plants. And then we can, look at change or return of those pollinator guilds over time following restoration.
Maybe just to finish up before we break and everyone can have a general mingle and chat, are there any direct questions to any of the panelists?
Oh, that's a good question. It's too hard to answer. Labour, reagents now with COVID. COVID has transformed DNA sequencing because there's so much competition all over the world for pipette tips and enzymes that we use. So it's not super cheap, but when you factor in the cost of, people in the field for long periods of time, it ends up, I guess, being quite cheap.
The thing that can make it a cost-effective tool is that it's a per sample cost rather than a per specimen cost or anything else. So if you have a mixed sample with 100 specimens or 100 species in it, it can be more cost-effective to just extract the DNA and sequence it rather than get, 200 specimens ID'd. And that's a big time saver there. So the more diverse your sample, the more cost effective in the end it can be. Yeah, and let's see. Also, when you're talking about fauna surveys and stuff, like weeks at a time, pitfall traps, long time. So you save on some time with the field sampling there because you're just collecting your samples and then going back to the lab. However, there are costs with, as Paul said, reagents and time in the lab and the analysis.
I guess there's also that depth of data that you get. So from one of Mika's pitfall traps where she's collected some arthropods, you can get, on that same sample, you can get information on the arthropod communities, you can get information on the plant communities they've interacted with, you can even get information on the vertebrate communities at the site. If you've got mosquitoes, for example, blood-sucking things in them, a lot of people in tropical areas use leeches to survey vertebrates. So we're trying to push towards that way where you just get multiple layers of data from the same sample.
Well, maybe just then, before we finish up and head off and have a mingle around the coffee and biscuits and whatnot at the back, just to illustrate that labour-intensive time point, and I'm sure that Holly can probably attest to this more than just about anyone else. Sophie, you determined the behaviour in one species in the Midwest over a 3-year period for the PhD. With the camera trapping, how many hours of camera trap footage did you use to come to your assumptions and evidence?
Hours of camera trapping footage was about 150,000 hours if you combine the camera trapping effort. But the real effort would have been walking those sites day in, day out, and I had a lot of time to think about this. But over the course of my entire PhD, I spent about 6 months on site, and I worked out in that time that it had walked pretty much the entire length of Italy by the time just to look at monitor lizards. So that shows how much I love them, but also how labour intensive that sampling can be.
So a little bit of a cost efficiency if we can send up one drone or one person out to collect some eDNA sampling. Right, I might invite everyone to just have a mingle and a conversation with Mika, Todd, Sophie and Paul and myself. And thanks very much for everyone coming along and pitching your questions and your thoughts.